Folks, if you were not here on Monday, you'll want to open up these slides to help understand what is going on for the DNA profiling worksheet. So we need to finish this up. We nearly finished on Monday, but just a reminder here, what we worked on Monday, we see that this individual is heterozygous. The number of repeats correspond to different peaks on our electropherogram, and we can calculate how common these traits are by using the formula 2pq if the individual is heterozygous. So it'd be this frequency times 2, or if they're homozygous, the formula is q squared. So it would be this frequency squared. These two alleles had different repeats, and so our formula is 2pq. These two alleles were the same, they were homozygous, q squared. So taking these formulas and using these numbers, we plug those in and we're able to figure out how common was the genotype at each locus. Now, this is where we need to continue. We are going to assume that each one of these genotypes are totally independent of each other. What you have at this locus has no bearing on what you would inherit on this other locus because they're on different chromosomes, so it would make sense that they're not connected. So once we have the probability of each locus, we can say, well, what are the chances to have all of these repeats? And that's the product rule. So take a moment, pause this video, multiply all of these frequencies together to see what the answer would be for number 6. Pause the video, do the math, grab a calculator if you need to. So I'm not actually going to show you the answer, but these are the frequencies above from A, B, C, and D. You multiply those all together, and I'm going to go ahead and just call that answer X. So if we wanted to testify in court to say there is less than a 1 in fill-in-the-blank chance that someone else has this genotype, to do the math, and I didn't really give you space, so we're going to do it down here. You take the inverse of X. So with your calculators, go ahead and take your answer here, divide it by 1, and write in the answer, and answer number 7. This is due today. When you are done with this, you are going to find your virtual biotech lab worksheet from last semester. We did part 1 and 2 already. So you are ready to complete part 3 and 4, all about PCR. If you've lost this worksheet, you can just click on this link here. Complete the virtual portion. If you'd like, you can just delete part 1 and 2. So you're starting here at part 3. But if you're doing the virtual version, please remember to attach it to Google Classroom. So for the links, all about PCR goes with part 3. Gel electrophoresis goes with part 4. If you did not do the starter, here is the slide. I probably should have started the video with this. And then when you are done with the virtual biotech lab, which is also due today, you can continue to work on your DNA test review. Make sure you bring those DNA release forms in on Thursday so we can do a buccal swab on Monday for our DNA extraction.